to be able to increase or decrease the response of the cell to that hormone. And we can do so by, if we were a neuron, and we had a neuron releasing neurotransmitters from the presynaptic neuron to the postsynaptic neuron, and we are creating graded potentials. Remember, let's say we're depolarizing the cell. The more channels we open, the more ions are going to flow into the cell, and the stronger the depolarization is going to be. We can increase that by having multiple neurons firing at the same time. Anybody remember what that's called? What kind of summation? Over time?
Spatial.
Over time is temporal. If we had, what you said is spatial. If the same neuron fires multiple times, then it's temporal. So we have multiple neurons firing, so we are increasing the number of neurotransmitters that are present. All right? We can use drugs to increase the effect by blocking their reuptake or by limiting acetylcholinesterase. If it's acetylcholine we're talking about, so acetylcholine isn't broken down as rapidly, or use drugs that mimic it. When we're talking about hormones and we're not addressing the pharmaceutical aspect of it, we can do the same thing. Here's our endocrine cell, well, a similar aspect. Here's our endocrine cell releasing hormones. And here's our target cell. If it is a protein type hormone, typically the receptor is going to be on the surface. And this is identified as a membrane bound receptor. And most of our hormones are protein-based amino acid derivatives or peptide hormones. And just a few of them actually have their receptors inside the cell. So there's quite a bit of a distance that has to be traveled usually between the endocrine organ and the target organ. So that means there's a greater distance for the diffusion and the dissipation of those hormones. So over here, we can double the number of neurotransmitters rather easily, and they're going to be trapped pretty close to the receptors to which they're supposed to bind. If we double the hormones being released here, not all of them are going to end up at the target cell, because not all the hormones that were made in the first place were at the target cell. So, some hormones are going to be in the blood and the brain and not at the ovaries, or at the foot and not at the ovaries or the testes. So it becomes a little bit more efficient to increase the number of receptors or to decrease the number of receptors. Now, if they're already made, if we have a pool of receptors already in the cell, then we can increase them by just having that in exocytose and put those receptors on the surface. So it will take a little bit longer if the cell has to actually synthesize them. Okay? But that increase in sensitivity, by increasing the number of receptors, is identified as upregulation. So even if we don't increase the number of hormones in the area, if we have more receptors that can bind that hormone, it's going to have a stronger effect on the cell. Now, the cell responds. We want that. We don't want to respond to that hormone anymore, but it's still there in the blood. And if we just wait for it to be excreted or to be broken down, it's going to take a while. And so the reverse can be done, and receptors can be removed from very quickly without having to break them down from the surface by endocytosis. Right? So decreasing the number of receptors is known as downregulation. Sometimes things become a little bit fancy here, and the hormone remains bound to its receptor. And rather than removing a receptor hormone in everything, a molecule made by the cell will come in and bind to it and block it from having any secondary effects. Okay, but we don't usually call that downregulation. Usually downregulation refers to the receptor numbers and sensitivity aspects.
Is that called like inhibition, or is that any kind of name, or?
Just what I put up there. Down and up regulation. Too easy?
What are you saying that was not referred to as down?
Oh, this one.
Yeah.
Yeah, inhibition. I thought you were talking about another name for that process. All right, so on the next page, I have some factors that can affect the time it takes to decrease the number of hormones that are circulating in the blood from a particular endocrine organ. We can certainly just get rid of them, okay? Excreting them into the urine or having them broken down by enzymes that normally do metabolic breakdown in the liver. Then products end up usually in the digestive tract. All right. They can be binding to plasma protein. Often, however, will protect them. So binding to a protein can sometimes have two effects. Here we have our hormone, and here we have the transport protein. That can make it so big that it doesn't get excreted in the kidney, but it can also make it a target for another cell that has a receptor for that will break it down. So again, there's variations, right? So the protein that it binds to can protect it from excretion, but can then make it recognizable for removal by other organs. So that's the difference. If you look on your lecture notes on page 7, were factors that lengthen, so you can see binding to plasma proteins versus attaching them to water-soluble molecules can make them easier to excrete into the kidney. So it just depends what type of molecule they're bound to, right? Whether there's a receptor for that molecule or not. So normally that would be part of our kind of introduction to hormones as a group. And now, any of you guys ever use clickers? I used to. This little I down here means that this is a clicker question. And if you guys have the radio little buttons, you can put A, B, C, or D as the correct answer, and then I can pop off a bar graph and show, that 60% of you got the right answer, and so on, how many people put whichever letter. The software has been updated, but it doesn't work with the questions I have on my PowerPoint slides. So I typically just put the questions up there. And I use these clicker questions two ways. In this area, this way, we haven't talked about these different types of hormones yet. And so I use this to introduce a new concept, expecting that you would guess at the answer, but it's making your brain work, making you think about it, all right? Or I would use it as a review. So when we first come back from a lecture, I might show what were clicker questions to review. With the clickers, everybody gets to respond instead of the people who actually say something, and the others are thinking in their minds, but they have to commit to an answer with the clicker. So I do miss them, but I still have the questions. Anybody recognize whether this is a lipid, a protein, or a carbohydrate molecule?
Lipid.
A lipid. Okay, anybody recognize what it's closest to? Cholesterol. So of those types of hormones that I have up on the board, which is derived from cholesterol? Steroids. Okay. The others are, we have 3 basic groups of hormones classified by molecular structure. Amino acid derived and peptides and glycoproteins fall under one. Okay. Well, actually, amino acid derived is one. peptides and glycoproteins is another, and lipids is a third. So we have two of the three groups are proteins, the third group being a lipid. You have this chart on your lecture note, fortunately, because nobody can read it up here except me. But if we look at these classes, and they help us to understand where the receptor would be located and how they're processed. You don't need to know all these examples primarily. I might ask for that on the homework just to get you to look at this chart. But certainly we've been talking about T3, T4. So you need to know that that comes from tyrosine. All right? In this unit we'll be talking about LH and FSH and estrogen and progesterone and testosterone. So by the time we're done with this unit, you'll know what type of hormones those are. But you don't have to go through and memorize that the parathyroid hormone is something or the H1 natriuretic peptide is another type. We'll be talking about that level of specificity when we get to the cardiovascular system or digestive system. So with that in mind, I do want you to recognize by structure a particular group, as in that previous diagram. So recognize an amino acid, a peptide, an icosanoid, and a steroid. So as we look at those three groups, we have the amino acid derivative. What I'm putting on the board is what I think is important on that slide. Okay? For those of you that haven't had me before and you're feeling overwhelmed by all the content to go through, what I test on 99.7% of the time is what I say in class and what's in your lecture notes. Okay? If it's not in those two areas, then it's not I may say it for, I might talk about it for clarification and then say, kind of like I've done with the adrenal cortex and the zones and stuff. Those aren't details. I'm saying them to help you explain something. Otherwise, if I don't say that, then anything I say in class and what's in your lecture notes is game. Okay. There's a lot of other stuff in your textbook, but more than we can cover. And I will probably cover some things that aren't in your textbook. Okay, but at least you have that definition. If you've covered what I say in class and what's on the lecture notes, you don't have to worry about some other surprise factor. So amino acid derivatives, we have two amino acids. Tyrosine. And from neurotransmitters and from lecture today, you're already familiar with T3, T4. All right, so this is a, recognize up here what we've talked about. And epinephrine, norepinephrine. They're very similar to each other, and I'm going to abbreviate them. Norepinephrine, of course, is a neurotransmitter. From the adrenal medulla, both norepinephrine and epinephrine are secreted into the blood. You typically don't have epinephrine coming from regular neurons. And then dopamine, which is our releasing or inhibitory releasing factor from the hypothalamus. The other amino acid is tryptophan. And that's used in making melatonin. Coming from the pineal gland. What gland would this be closely related to by location? Pituitary. So pituitary grows from the floor of the third ventricle. Pineal gland from the roof, posterior roof, the back wall, if you will, of the third ventricle. This is the circadian rhythm, 24-hour cycling. You can buy forms of melatonin at the drugstore if you want to try to get back into a regular sleeping cycle or you travel a lot and get jet lag. I've never tried it, so I don't know. I don't know when you would even take it. Beginning, end? I don't know. So these here. All right, then we have peptide or protein hormones. So these are going to be bigger. And we have glycoproteins. Okay, and then just small and large peptides. Just don't have glyco means it has the carbohydrate groups on it. And again, there's a list here of short ones and large ones. Diagrammatically, you were to recognize that you have a diagram on the next page of your lecture notes. So here's the single amino acid one, here is a short peptide, here's a larger, longer amino acid ones. So you would recognize, obviously, these as steroids. Okay? They don't have that ring shape. So if you have a long sequence of amino acids, it's going to be more than 1 or 2 modified amino acids. So you don't have to recognize a particular hormone's molecular structure, but if I give you a diagram of one, be able to classify it into one of those 4 groups. Okay. And you have a similar diagram on the next page of your lecture notes. So these are all both amino acid components, and the last one is lipid. We've already talked about cholesterol. All right. The other group are derived from a fatty acid known as arachidonic acid. And it looks like that. So it has an open chain structure. Of course, there's oxygen and hydrogens and stuff that aren't on there. Arachidonic acid is further modified into groups of molecules that fall under the general classification of icosanoids. Other than prostaglandins, which are produced by both females and males, and they don't come from the prostate gland either, for that matter. That's about the only one of the icosanoid types. There's PGE and a bunch of different kinds of prostaglandins. Prostaglandins cause the uterine wall muscles to contract. All right, so they're a component of initiation of labor. The other ones we'll talk about with the blood and the immune system are thromboxanes. This helps platelets be sticky. And with white blood cells and their immune components, leukotrienes. Oh, like really strange words.
Honey, we're out of leukotriene.
I'll have to stay home sick today. Then we have cholesterol, which is the 4-ring structure. And that makes steroids. So estrogen, progesterone, testosterone, we'll be covering those. Not so much cortisol, but certainly the gonadal hormones in this unit. Now, of that grouping that we have up there, 2 major groups and 1 smaller hormone are considered to be lipid soluble. All right, so obviously arachidonic acid and cholesterol is lipid soluble. So if they are released from a cell, they don't need endocytosis. They can just diffuse through the membrane. And where would you expect to find their receptor? On the surface of the cell or inside the cell?
Inside.
Inside the cell. All right. So they're going to have their receptor either in the cytoplasm or in the nucleus. And as we'll see tomorrow, even if it's in the cytoplasm, it's going to end up in the nucleus once the hormone binds to it. Okay. So it has to be lipid soluble. Now we talked about T3, T4 are lipid soluble enough that they're not soluble in water, but they still need transport help to get across the membrane. But their receptor is inside the cell. of these are membrane bound. So they're out on the surface. We need some form of communication between that surface of the cell and inside the cell. And that's going to lead us to the next 20 minutes or so of content. How does that happen?
Lipids are non-membrane bound.
Now,
Non-membrane bound. Lipid bound.
membrane bound receptors, we're not going to cover all of this today. We're just going to get to the introduction of the common pathway of G proteins, and we'll finish the rest of this up tomorrow. But let's go back to the nervous system. Can I erase this image here?